My lab studies virus-host interactions in gamma herpes virus pathogenesis. Gamma herpes viruses include the human viruses Epstein-Barr virus and Kaposi sarcoma-associated herpes viruses. And these are viruses that really infect cells in the immune system, so primarily B cells. And by infecting those cells, they can turn those cells into cancer cells. So once you have a herpes virus, you keep it forever. And the fact that we are chronically infected with these viruses, specifically in the B cells, predisposes those cells to becoming lymphomas down the road. So the main question we were interested in asking is, are there any intrinsic defenses that those B cells have that will protect a cell that's being targeted by a virus and restrict latency establishment, chronic infection of the cell, and then down the road, tumorigenesis caused by the virus? So we focused on a really important tumor suppressor molecule, P53, and we simply asked, using a mouse model of gamma herpes virus infection, whether P53 was activated by the virus during colonization of the host, and whether P53 played a role in restricting chronic infection by the virus. And the answer to both of those questions turned out to be yes. So we used that preliminary data to write a new R01 grant, and the National Cancer Institute liked it and they picked us up. So in terms of what benefit this will have for UAMS, the UAMS community in general, I guess right off the bat, it provides resources so we can train future doctoral students, master's students, and postdoctoral fellows. And then in the long term, we hope the information that we gain through our project will help us to understand the ways that gamma herpes viruses can cause infection, and then let us understand whether we can use these, harness these tumor suppressor pathways in such a way that you can help eliminate gamma herpes virus infections or reduce the pathogenesis and disease caused by the viruses. In terms of impact on the field, there's really not much known about host proteins that provide intrinsic resistance against herpes virus infection, especially not in using in vivo models. So in doing this, we're providing basic new information in terms of how the host responds to infection, and in particular, which proteins are important for limiting chronic infection and disease caused by the virus.